Well, we are so happy today to have Dr. Ksenia Kastanenka with us. So she is an Assistant Investigator in the Department of Neurology here at Massachusetts General Hospital and is also an Assistant Professor of Neurology for Harvard Medical School. Her research interests lie in the sphere of aging with a particular emphasis on Alzheimer's disease. Her laboratory is focused on studying the circuitry disruption during the disease progression and mechanisms of action of Alzheimer's therapeutics aimed at reversing the disease. So we're really excited to have her here to tell us a little bit more about preclinical therapy development.
Yes, Kelsey, thank you so much for coming over and for all the hard work that you do at Alzheimer's Association. I am happy to answer any questions you have. I am an assistant professor here at Massachusetts General Hospital and Harvard Medical School, and my laboratory is mainly focused on preclinical research, so that's my area of expertise. And any questions you have, I will be happy to answer.
Great. So why don't we kick it off by having you tell us a little bit more about what preclinical research for Alzheimer's therapy entails?
Sure, of course. So we preclinical researchers are actually part of the bigger team, as I'd like to call, and our goal is to cure Alzheimer's disease. As all of you know, there's currently no cure for Alzheimer's disease, and we're focused to changing that. And us as preclinical researchers, we have a very specific mission. We have a very specific job, and that is different from the clinical work that you're probably most familiar with, where drugs are being tested in Alzheimer's patients and healthy individuals. And actually in the preclinical space, we screen a number of different compounds. We test them in cell culture models, in animal models, to see which compounds have the most promise before they go into the humans. So that's what we are mostly focused on.
Yeah, that's really interesting. And so what are some of the brain targets that these new therapies are being developed to target?
Right. So Alzheimer's disease is the major cause of dementia, and the memory center of the brain is hippocampus. So that's actually so a great therapy would target that brain region. Also, there's cognitive decline, and neocortex is mainly responsible for cognition. So a great therapy would also target that brain region as well. Also, as part of the disease progression, there's a number of other brain regions that are affected. So an ideal therapeutic would be able to target each one of those brain regions.
And so just to set the stage, why don't we hear from you a little bit more about what's actually happening in the brain with Alzheimer's disease, some of what the pathology is?
Of course. So Alzheimer's disease is a neurodegenerative disorder, and it's progressive. That is, there's changes happening at each stage of the disorder. So if we are interested in coming up with a therapeutic that's going to be effective, we have to be able to interfere at the right stage with the right therapeutic. So what Alzheimer's disease is characterized by major hallmarks such as deposition of amyloid plaques that are extracellular in the extracellular space of the brain, and neurofibrillary tangles, which are intracellular and occur inside the neurons. There's also a substantial neuronal cell death. In addition to these hallmarks, there's disruptions in neuronal circuitry. So the memory centers of the brain, as well as the cognition, they're maintained by the circuitry in the brain, and there's disruption of different connections in the brain. So that's actually something something that we're very interested in studying.
Why don't you tell us a little bit more about your research looking at neuronal circuits?
Yes, of course. So my laboratory is geared, the effort in my laboratory is geared twofold. The first is we collaborate with industry leaders and we test their promising therapeutics that are either in clinical development or in the clinic already, and we bring those therapeutics in and we test them in our animal models of Alzheimer's disease. The second part of my research actually has to do with memory disruptions and sleep disturbances. So there's a certain brain rhythm that is called slow oscillations that is important for consolidation of memory during sleep. And it's really interesting for us to study because we found recently that there's disruption in the activity of the circuitry that underlies that brain rhythm. And we found this in an animal model, and this is actually parallel to what is happening in Alzheimer's disease patients. Early on, during early stages such as MCI, mild cognitive impairment, or early Alzheimer's disease, the patients are not able to fall asleep and sleep well, in addition to their memory impairment. So we're interested in studying these slow oscillations to see if we can understand how the circuitry gets dismantled and how we can restore its activity to normal.
Yeah, that's really interesting and really important. I'm sure many of the caregivers that we speak with, they observe sleep impairment in their loved ones, and so being able to better understand that process would be very helpful.
Great.
So you've mentioned animal models a few times, and so can you tell us a little bit more about the animal models that you use and kind of what pathology they may have?
Yes, of course. So we believe that animals are a great way to try to ask some of the questions that we're not able to ask in a patient, for example. So the scientific manipulation that we can perform in a human patient is limited. So in that case, if we're trying to understand the causes of Alzheimer's disease or how Alzheimer's disease progresses, animal is a great place to go. So we are in neurodegenerative field, specifically Alzheimer's disease. We're actually very fortunate because we have the genetics that we can use, the genetic mutations that we can put into an animal and make a great animal model of Alzheimer's disease. So we're able to use those models that recapitulate some of the pathology in the human and study how that pathology progresses. We're also able to use therapeutic to try to reverse some of that pathology as well.
That's really interesting. And so you've mentioned brain stages clinical stages before. And so what stage of Alzheimer's disease are some of the therapies you're developing targeting? Are they for all different stages of the disease progression, or are you focused on one individual point in time?
Right. So you're probably aware there's relatively recently there have been a great number of clinical trial failures. And when later stages of Alzheimer's disease are being targeted, perhaps we're not at the right time point to interfere with the disease progression. So there's been a shift to target earlier stages of Alzheimer's disease, early AD. So actually aducanumab that recently failed in phase 3 clinical trial was targeting that early stage of Alzheimer's disease. So it looks like we should be able to go even earlier, perhaps MCI stage, the mild cognitive impairment, or even before the symptom onset. And there's actually a great research effort that is currently underway to try and interrogate the disease at these early stages.
That's great. And so let's shift gears a little bit. And so you've mentioned that your team looks at some of the therapies that are being developed. And so what actually happens when a new therapy is being investigated, and what needs to happen in order for it to get into clinical trials?
Right, of course. So what usually happens is initially there is an idea or hypothesis that's being tested, and there's all these different therapeutics that undergo a screen. Right, so we start with a great number of potential compounds, and that screen happens in something like a cell culture, for example, in a cell line. And then the most promising compounds enter the animal studies because animal studies take longer and it's not as efficient of a screen. So these compounds enter the animal models, and the compounds that are actually able to cure Alzheimer's disease in an animal, those are the compounds that enter the clinic.
Oh, interesting. And so are there specific criteria that need to be met in order to make that shift? from animals to humans?
I would imagine there is. I'm not really that familiar with that line of work, but I would imagine there is a very strict criteria that needs to be met in terms of safety, in terms of efficacy before entering a human trial. And of course, there's the FDA regulation that needs to be satisfied as well.
Yeah, no, that makes sense.
Yes.
And so you said that if the new therapy looks to be effective and reverses changes in animal models, that's what would be taken into human studies. And so what would some of those changes be? Are we looking at mice that have amyloid beta in their brain, and you're also looking at changes in their cognition? How does that really work?
Right. So I can give you an example of aducanumab that recently failed a clinical trial, but it's a great antibody, it's a great therapeutic to try, and that's why it has gone so far as to the third stage within the clinical trial. It is anti-A beta antibody, and we actually worked very closely with Biogen on this antibody, and we tested it in our animal model. So our animals developed amyloid plaques, they developed amyloid, and also they had calcium overload within the cortical neurons. And calcium is very important not only for your bones, but it's also very important for neuronal health. And if levels of calcium are too high within the neurons, then the neurons become unhealthy. So what we did, we tested this antibody to see if it clears amyloid plaques, which it did, and whether it restores the calcium within neurons, which it did. So that's great. And that paired together with the preclinical data at Biogen itself, where they saw clearance of amyloid plaques, in the animal model, that allowed that antibody to enter the safety trial and then the efficacy trial as well.
It's a really interesting kind of process, and it makes a lot of
Exactly.
sense. And so you've mentioned a few different ways that you've been screening these drug targets. Can you tell us a little bit more about some of the other techniques that you may be using?
Sure.
In your lab?
Sure. So in my laboratory, we're actually using really cool technology. We are using multiphoton microscopy, which is a high-resolution imaging technique. It allows imaging individual neurons over time in living, breathing animals, which is really remarkable. So we're able to see what happens to the brain as the disease progresses, for example, or what happens as part of a therapeutic regimen. So we're able to dose the animals with the drug and see if they get better. And multiphoton microscopy allows us to visualize some of those structural as well as functional changes in the brain, which is really remarkable. Another amazing technology that we use in my laboratory is called optogenetics. So optogenetics is that technique that took neuroscience by storm just several years ago. It's a relatively new technology, however, it's very widespread at this point. And the reason is because we're able to non-invasively activate specific neuronal populations. And that is really important when trying to understand how circuitry assembles, how it disassembles as part of the disease progression. So these are really amazing leading edge, cutting edge tools that we use to try to understand what happens in the brain.
Yeah, that's really exciting. And so you've mentioned neuronal circuitry a few times. Do you mind explaining to our audience a little bit more about what that means?
Yes, of course. So our brain is basically made up of different cell types, different cells. So just like in the blood there's red blood cells, in the brain there's neuronal and non-neuronal cells. Neuronal cells are thought to be responsible for cognition, for memory, for us to be able to see things, perceive things. And these neurons are connected to each other. So those connections is what is actually being disrupted as part of Alzheimer's disease. So if neurons are not able to communicate with each other, if I'm telling you something and you're not hearing me, that means that our circuitry, we're not connected. And that's essentially how circuitry relates to Alzheimer's disease. And that's what we're trying to reverse or restore. We're trying, if For example, a neuron is active during a period of time when it shouldn't be, that's a problem. Or when a neuron is silent when it should be active, that's also a problem. So we are interested in restore the normal function, the neuronal function, so it does fire when it should fire, to be able to maintain the normal processes.
It's really interesting to connect the biology of the disease to the behaviors and the symptoms that we see of the disease as well.
Exactly, and that's what we are interested in, trying to understand what happens in the brain to be able to alleviate the symptoms and reverse the disorder.
So while we're on that topic, why don't you tell us a little bit more about your research involving sleep and the circuitry that's going wrong in the brain with the sleep processes?
Yes, of course. So I mentioned that there are sleep disturbances in addition to memory disruptions in Alzheimer's disease patients. And we also see this in our animal models. So it was really serendipitous discovery that we made several years ago now, where we were visualizing the activity, the brain activity, this brain rhythm in our animal model of Alzheimer's disease, and we saw that the brain rhythm is totally different from that in healthy animals. So there was something wrong. And slow oscillations are really important for the consolidation of memory during sleep, as I mentioned. So we're really interested in trying to figure out why are slow oscillations disrupted? What is the mechanism that underlies that disruption? And then can we restore that brain rhythm back to normal? And that's something we've done using optogenetics in the animal. And there are actually several reports in the literature to suggest that this could be efficacious in humans as well. There was actually recently a report to suggest that elderly individuals have disrupted brain rhythms as well. And if in humans, if we restore their activity to normal, they perform better on a memory task, which is really amazing. So if we can take that into Alzheimer's disease patients and see if we can interfere in that regard, that would be really amazing.
Yeah, that would be. And so we're thinking about sleep in the context of Alzheimer's disease. Do you think that sleep has a role in how the pathology progresses, or do you think it's more of a side effect of the pathology?
So that's really amazing question, and I don't think anybody knows the answer to that. So that's the chicken and egg question, right? What goes wrong? Is it the circuitry disruption that is part of the disease progression that causes sleep impairments, or is it sleep impairment that causes the circuitry disruption? And there are experiments that we can perform that are currently underway to try to answer this question, but it is still early days.
Mm-hmm. What would be some of those avenues of research? I feel like our audience would probably be interested in learning more about that.
To try to
to
restore
kind of elucidate kind of what those differences are.
Yes, exactly. So we can try to study the circuit and see how it's disrupted, and see whether it correlates with sleep disturbances, and we can actually manipulate the circuit. We can restore its activity and see if sleep patterns normalize. So that would be a really great way to do that. Or we can modify sleep. So we can sleep deprive animals, for example. So I have a toddler at home, and I've been sleep deprived for a number of nights now, so I know exactly what it's all about. But if we do something like this in an animal, can we disrupt the circuitry in a similar way that we see in Alzheimer's disease? Those types of experiments could be possible.
Yeah, it's really interesting. So shifting gears a little bit more back to preclinical research now. So you've talked a lot about therapies for amyloid beta. Are there therapies in the preclinical stage focused on other targets of the disease as well?
Yes. So amyloid beta has been the major focus. And and that is due to amyloid hypothesis that has been a prevalent hypothesis in the field. There's also tau that is being targeted. So there's a number of different compounds that are currently in preclinical development to target tau. And tau is a great target. There's also ApoE. So ApoE4 is a risk factor for Alzheimer's disease. So there are several ideas about how to manipulate ApoE and try to restore it to a more healthy conformation.
Yeah. So it seems like there's quite a few targets involved.
There are. And that's one of the amazing things about preclinical research. We're never out of ideas to try. And that's why I think there's great hope for Alzheimer's disease patients, because we're going to keep trying until we get it right.
That's really exciting. I think that's really important to keep in mind, too, that there is so much research happening at every stage, really focused on identifying the best treatments for people living with dementia.
That is exactly right. And like I said, we all have one goal inside, and that is to cure Alzheimer's disease, whether it's with this type of therapeutic or this type. At the end, it doesn't matter. It's as long as we get to that finish line.
Mm-hmm. Do you think it will end up being more of a combination therapy, or do you think it will depend on when we're targeting the disease?
So Alzheimer's disease is one of those disorders that we thought we're going to figure out relatively quickly. It seemed pretty straightforward. There's amyloid plaques, there's tau tangles. If we can minimize those, perhaps things get better. And over the years, we understood that it's a very complicated disorder. So depending on the stage of the disease and the more progressive the disorder is, the further you are in the disease progression, the more disruptions are there in the brain circuitry, and the more issues there are. So at these later stages, perhaps combination therapy is reasonable, but it does have its own hurdles.
Mm-hmm. That's really interesting to kind of think about all of the different targets and whether it's a combination or if we need to be better at targeting at a certain stage. Are you familiar with the biomarkers that are out there, or do you know of any ways that we can detect some of these pathological changes while someone is going through the disease?
Right, of course. So until recently, the only way to diagnose Alzheimer's disease was through a test.
Mm-hmm.
A patient comes in to a neurologist's office and they give them a battery of tests, and depending on how they score, they determine whether they have Alzheimer's disease or not. And then autopsies reveal that perhaps this is not the most efficient way to go. So now, with help of MRI, with PET imaging, we are better positioned to use some of those biomarkers to diagnose Alzheimer's disease. So that's been extremely helpful. So there's PET imaging for amyloid, which has been developed a number of years ago, but is more used mainstream. There's also development of PET imaging for tau. There's also CSF biomarkers. We're able to measure A beta and tau levels in the CSF, which is really incredible. And there's a great development in that regard as well. And also functional connectivity is also very important. So each one of those is a great tool to use.
Yeah. Do you think when you mention functional connectivity, do you mind elaborating on that a little further?
Right. So there's great work by Risa Sperling on the default mode network, where she saw that there's certain brain regions that are usually active during the normal functioning. They're not active on Alzheimer's disease, or there's regions that are hyperactive but should be silent at that time, and that's mainly done with MRI. fMRI. So that's definitely a great line of work.
So a question that just came in was someone asked a more personal question about your research and how long you've been involved in Alzheimer's research. Has this been the field you've always been in?
No, actually. I got involved with Alzheimer's disease in 2011. So I have been in the field of Alzheimer's disease for 8 years now. That's once I started here at MGH.
Oh, great. While we're on that topic, what led you into this field of Alzheimer's disease research?
Of course. So I received my Ph.D. training in neuroscience at Case Western Reserve University, where I was trained in very basic science. So I was trained how to do science well. And at that point, my line of work was very different. Towards the end of my training, I really wanted to be involved in more translational research, particularly Alzheimer's disease, because I knew there was a great need to develop therapies, and I became very passionate about it as a result. And now every time I come into the lab, that is my goal. I want to forward the field, contribute to the field, so that there is a cure. So that's what drives me every day.
Yeah, that's really exciting. I think it's really important to have that motivating factor.
Absolutely,
sure.
Because I see a lot of people suffering,
Mm-hmm.
and there's nothing that can be done. So it's definitely a great purpose.
Mm-hmm. So a question that we often hear is, how close to a treatment are we? So that's really hard to predict, but interested in what your thoughts are looking from the preclinical research perspective.
So we were hoping we would be there by now. There have been great successes in MS field, multiple sclerosis, by Biogen, and we thought we would get there with Alzheimer's disease. I do hope it's any day now.
Mm-hmm.
I do hope it's any day, but we're not there yet.
Mm-hmm. Are there clinical trials looking at lots of these different targets, or are they more focused on one type of pathology in Alzheimer's disease?
Right, so there's a lot of clinical trials that are focusing on tau now. So I think A-beta is out of favor now, so there's more focus on tau and see if we can interrogate the disease from that perspective. So we'll see how those trials go.
Yeah, it'll be interesting.
Mm-hmm.
And I know that I've seen recently that we have a lot of shots on goal. So there's a lot of clinical trials happening at different phases and focusing on different targets, which is exciting to see.
Right. Exactly.
So you mentioned earlier talking about targeting individuals before they may even have symptoms.
Mm-hmm.
Could you tell us a little bit more about that research?
That would be incredible if we could do that. So that's actually something that I am interested in my work with animals. We're trying to study animal models of Alzheimer's disease early in the disease progression, before even deposition of amyloid beta, which is itself a very early stage of Alzheimer's. So it's definitely an important time point to study. However, it's very difficult to do that in humans, because unless you're troubled, unless you have some sort of symptoms, you will not necessarily be asking for a PET scan from your physician. So it might be a challenge to try to interrogate those early disease stages. However, one way to go about that is to try and recruit subjects that are at risk for Alzheimer's disease. So I mentioned APOE4 genotype, and there can be a genetic screen done. It doesn't matter what your age is, and determine whether you're an APOE4 carrier. And if that's the case, you'll make a great candidate for a research study.
So while we're on the topic of participating in clinical studies, why don't we talk a little bit more about how a clinical study works? Could you tell us a little bit more about the phases of clinical trials and what those mean?
Sure. So there are typically three 3 phases of clinical trials. The first is usually a safety trial, and then second phase is where the drug is actually tested in a small population of Alzheimer's disease patients, just to see if there's if it's efficacious at that point. And then phase 3 study is a huge study where a great number of subjects are recruited to see if it works in the general population.
It's interesting to kind of see how they study it to make sure that any therapy is safe and effective for the general population.
Exactly.
It's really important.
Exactly, yes.
I know it's frustrating that it takes so long for these treatments sometimes, but it's important that we're doing it carefully.
It really does. Although there are several mechanisms that pharmaceutical companies can try and take advantage of to expedite the development of therapeutics. And actually, FDA recently relaxed the guidelines for drug development for Alzheimer's disease. So that will also help in therapeutic development space.
Yeah, that definitely will. So thinking back to your research on sleep, so you mentioned earlier how consolidation of memory may happen during sleep. Do you mind explaining a little bit more about that topic?
Yes, of course. So as we go about our day, we have different experiences, and some of those experiences we will remember for a long time, like your wedding day, for example, or some of those experiences we forget right away, like what I had for breakfast. And the consolidation, the memory storage for long term happens during sleep, believe it or not. So as we are relaxing in the hours of the night, that's when the magic happens. Actually, our brain is very active while we sleep, and during those deeper stages of sleep called slow wave sleep, there are these rhythms, the slow wave rhythm, hence the slow wave sleep, that drives slow oscillations, and that's when consolidation of memory actually happens. And it's really remarkable to see that that's the brain rhythm that is disrupted in Alzheimer's disease patients.
Yeah. So why don't you explain a little bit more what a brain rhythm is and how would we detect that? How can that be measured in research?
Sure. So there are a number of different brain rhythms. So brain rhythm is basically, if you think about an ocean wave, so waves come in to the shore, right? So that's what a brain rhythm is. And there's a certain frequency, right? So there's slow frequencies, such as slow oscillations, which occur at less than 1 hertz. And then there are faster brain rhythms. And one of those is gamma brain rhythm. So slow oscillations are important for memory consolidation during sleep. And gamma is actually important for learning and absorbing information. So interestingly, there is research that's been recently done at MIT where they see disruptions in gamma oscillations as well. So we see that the brain rhythms are extremely important for normal function and are disrupted in Alzheimer's disease.
Oh, interesting. It'll be interesting to see why that happens. Exactly.
So we believe that some of these toxic proteins, such as A-beta, might have something to do with it, and that would be a great way to try to interfere with the disease progression.
Yes. And so can we measure these brain rhythms in humans as well, or are they only measurable in animal models?
Absolutely.
Right now?
We can measure these brain rhythms in the animals, and we can also measure them in humans using EEG and electroencephalography. So it's definitely something that we can monitor in humans as well.
Do you think that could ever be used as a way to predict who may be at an increased risk of developing dementia or Alzheimer's disease?
That would be incredible if we could use brain rhythms as a biomarker. That would really be something. Yes.
Yeah. So how do you study brain rhythms in your lab, or are you more focused on circuitry that kind of leads to those brain rhythms?
Both. So we're interested in studying individual neuronal and non-neuronal neuronal cells that make up the circuit that drives the brain rhythm. And also we're looking at the global activity within the brain to try to see what these brain rhythms look like and how they're disrupted.
I think it's been really exciting to kind of pull back the curtain on how a lot of this research happens and what the preclinical stage looks like. So you mentioned cell cultures before and how that's a tool that you use and a model that you use in your lab. Can you explain to our audience a little bit more about what that entails and what those cell cultures really are?
Of course. So there is a number of different cell cultures that we can use. Some of those are cell lines that have been passed, and some are, for example, primary neuronal cultures that are isolated fresh, for example, from animal models. And we're able to grow these cells, which can be neuronal or non-neuronal cells, in a dish. And they're happy, they survive for days, weeks, and then we're able to study them, right? So we're able to manipulate their genetic makeup of these cells. We're able to measure their calcium, measure their ionic balance. We're able to see how these cells respond to a number of different insults, such as amyloid, which we know is toxic, or tau. We're able to see if these cells perhaps send the proteins between their synaptic connections, as we've seen for tau. So it's a powerful tool to use these cell cultures. And one interesting development, fairly recent development, is actually we're able to culture human cells. I'm not talking about stem cells. I am talking about taking a skin cell and differentiating it into a neuron. So we can take a skin cell and we can make it into a neuron, and then we can study it. So we can study your specific cells to see how you, as an Alzheimer's patient or somebody who is at risk for Alzheimer's disease, how you would respond to different drugs or manipulations, which is really incredible. This is the future of personalized medicine.
Yeah, that's really incredible to think about, and that's such a powerful technique available.
Oh, yes.
So why don't you just mentioned personalized medicine, so why don't you tell us a little bit more about what you mean by that?
Right. So what I mean by personalized medicine is being able to take tissue or cells from a patient and study those in a laboratory and come up with just the right therapeutics that would allow these cells to respond. So in the case of cancer, some cancer patients respond to certain therapies and some don't. So if we can culture the cells in a dish, in a laboratory, and see which drugs the cells would respond to, then we're better positioned to target the therapy to specific patients' needs.
That's really interesting, and that really sounds like a way that we'll be able to find better treatments for different people, since we know everyone is different, and so being able to more personalize it is really important.
And you're actually hitting on a very interesting point, because we think about Alzheimer's disease as one disorder, but what if it's actually different disorders, right? So that's something that we're actively thinking about as well.
Mm-hmm. So what are some of those studies that are happening to differentiate what type of dementia someone has, or what those small differences at the biological molecular scale between different patients might be?
So one way to look at the differences would be by looking at the genetic makeup, right? So we know that certain mutations in the amyloid precursor protein gene that results in production of amyloid beta, the toxic protein, or some of the mutation in the presenilin gene that leads to production of amyloid beta. Some of these mutations lead to early onset Alzheimer's disease. and I mentioned APOE4 genotype as well as a great risk. So genetics are a very powerful way to try, to understand the disorder a little bit. Although familial Alzheimer's disease is a small percentage of Alzheimer's disease, so there's a great number of the genome-wide screens to try to pinpoint some of the other genes that might tell us a little bit more about the disease.
It's definitely a complex disease, and
yes, it is.
It's really incredible that we have all these techniques at our disposal, and all of these different techniques that we can use to really try to find and identify the best treatments.
Exactly.
And so I think we're almost out of time. So is there anything else you'd like to share with our audience on preclinical therapy development, what hope you have for the future?
I really hope that next time we meet, we'll have something closer to the market, if not on the market. That would be great.
That would be great.
That would be great.
Well, thank you so much for your time. We really appreciate it.
Thank you so much for coming over and visiting me at MG.
Yeah.